Chapter 9, Cellular Communication. Let's discuss intracellular signaling, which is communication between cells, and intracellular signaling, which is communication within cells. When we talk about communication, we worry about ligands, which is released by individual cells. They're small and often volatile, and they interact with target cells. Those target cells have proteins that are known as receptors. There are many different types of ligands, and there are many different types of receptors. We'll discuss many different examples. There are 4 forms of chemical signaling. In autocrine signaling, a cell targets itself. In signaling across gap junctions, a cell targets another cell that it's connected to by a gap junction. In peregrine signaling, cells talk to neighbors, and in endocrine signaling, cells target distant cells, and they communicate with one another via the bloodstream. We'll look at all 4 of these types more closely. In peregrine signaling, we're looking at local communication between neighbors. There's movements of signals via diffusion, and the responses tend to be very quick and very short. The ligands, once received by the intended cell, are almost immediately degraded, which helps to rebuild their potential chemical and electro gradient, allowing a message to be shared quite quickly. The difference between the presynaptic cell and the postsynaptic cell is called the synaptic gap. It's very small and allows for the diffusion of signaling molecules like neurotransmitters. Enzymes in the synaptic cleft degrade some types of neurotransmitters that terminate signals. This is an example of peregrine signaling. If it doesn't sound familiar, this is what's happening in your nervous system that allow your nerves to talk to one another. Endocrine signaling is signaling from a distance. These originate in the endocrine glands and have slower responses but longer-term effects. The ligands in this situation are often hormones that move via the bloodstream and regularly exist in our bodies in low concentrations. Here's an example of endocrine signaling if you would like to see it in image form. There are secreted molecules that diffuse into the bloodstream and trigger responses in target cells anywhere within the body. The third type of signaling is autocrine signaling, and this is self-signaling. It happens a lot in developmental control and even apoptosis, which is programmed cell death. We'll discuss that later. Autocrine messages can affect neighboring cells if the signal is strong enough. Here's an example of autocrine signaling. Secreted molecules diffuse locally and trigger a response in the cells that create them. Finally, we have direct gap signaling. These are connections between neighboring cells. The connections are known as plasmodesmata in plants and gap junctions in humans. Very, very small signal molecules are shared this way, commonly calcium. There's an immediate reaction, and it allows for processes like the coordination of movement to occur quickly and easily. There are 2 main types of cellular receptors that take in all these messages. Internal receptors are also known as intracellular or cytoplasmic receptors, and they respond to simple hydrophobic ligand molecules. They're capable of traveling through the plasma membrane because of that hydrophobia, and they easily affect gene expression. In fact, the ligand and receptor can move together into the nucleus to initiate or block gene expression. Here's an example of a hydrophobic signaling molecule diffusing across a plasma membrane and interacting with intracellular receptors in the cytoplasm. Many intracellular receptors are transcription factors that interact with DNA in the nucleus and regulate gene expression. Now let's talk about cell surface receptors, or external receptors. These are often transmembrane receptors. They are anchored internal proteins that bind to the external ligand molecules. They span the entire plasma membrane. The ligands that react with cell surface receptors don't have to enter the cell, and they exist in 3 general categories. They are ion-linked, G-protein-linked, and enzyme-linked receptors. This is an example of an ion-linked receptor. They bind to the ligand and open their channel. These receptors have extensive membrane-spanning regions. There are amino acids within the hydrophobic regions next to the tails that are hydrophobic themselves, so they're quite happy where they're sitting, and that shared hydrophobia keeps them from moving out of the membrane. The channel itself, however, is hydrophilic on the inside, and that allows molecules passage. Conformational changes allows materials to move at a predictable rate across the plasma membrane. Gated ion channels form a pore through the plasma membrane that opens when those signaling molecules bind. The pore then allows the ions to flow into or out of the cell, as I mentioned, at a nice controlled rate, depending on the needs at that particular moment. This is the second kind of receptor, the G protein linked receptor. They bind to the ligand and then activate ion channels or enzymes. They generally have 7 transmembrane domains, and each has their own structure and binding sites. There are 5 steps to this process. Normally, the ligand is inactive and the G protein unit is kind of just hanging out on the plasma membrane. When the ligand binds, the G protein linked receptor releases GDP and picks up GTP. This big G unit then splits off into an alpha and a beta gamma unit, and either of those units can then go activate or share signals. Eventually, though, that alpha will be hydrolyzed and the beta gamma is going to be deactivated, and the whole unit will come back together and the G protein will reassemble. These features of G proteins mean that they are heterotrimeric. They have 3 subunits. When a signaling molecule binds to a G protein coupled receptor in the plasma membrane, a GDP molecule associated with the alpha subunit is exchanged for GDP. The beta and gamma subunits dissociate from the alpha unit, and the cellular response is triggered by either the alpha subunit or the dissociated beta-gamma pair. Hydrolysis of GDP to GDP terminates the signal. These G protein-linked receptors function in disease etiology. Bacteria that are pathogenic to humans can release poisons that interrupt their processes, leading to illnesses such as pertussis, botulism, and cholera. In cholera, for example, the waterborne bacteria Vibrio chloriae produces a toxin, chlorogen, that binds to the cells lining the small intestine. The toxin enters into these intestinal cells where it modifies the G protein that controls the opening of our chloride channels and causes it to remain continuously active. This results in an incredibly large loss of fluids from the body and potentially fatal dehydration as a result. Enzyme link receptors are cell surface receptors with intracellular domains, also known as ICDs, and these are associated with enzymes. The receptors themselves might actually be an enzyme, which helps to speed up the process. The enzyme-linked receptors normally have large extracellular and intracellular domains, but the little membrane-spanning region actually only consists of a single alpha helical region of a peptide strand. When the ligand binds to the extracellular portion, the signal is transferred through the membrane, activating some sort of enzyme. The activation of the enzyme sets off a long chain of events within the cell, and it will eventually lead to some form of response. The molecules that actually do the sharing of the signals can be quite different. Some are small and hydrophobic, and they are capable of diffusing through the membrane. Steroid hormones are an excellent example. They have hydrocarbon skeletons with 4 rings and various functional groups that can give them different properties. Steroids bind with carrier proteins when they need to move within the blood, and this even helps to keep them from being degraded unnecessarily. Some signals can be water-based and bind with extracellular domains, while other signals, like nitric oxide, function via diffusion and are known to relax tissues. These, in particular, have very short half-lives and don't exist in our bodies for long. Let's talk about how that binding actually initiates the signaling pathway. The ligand itself will bind to a cell surface receptor. The activation of the receptor's intracellular components sets off something known as a signaling pathway or signaling cascade. This turns on second messengers, enzymes, and activated proteins, which then go interact or talk to other specific proteins. These messages are shared in activated chain reactions that eventually lead to a physical change in the cell's environment. The events in a cascade occur in a series, much like a current flows in a river. Interactions before a certain point are referred to as upstream events. Events after that point are referred to as downstream events. These processes can get very complicated very quickly. Cellular proteins can affect various downstream events, and a single pathway can branch off towards different endpoints. In some instances, the exact same ligand can be used to initiate different signaling pathways in different cell types. Another complicating element is the concept of signal integration. This is when signals from two or more different cell surface receptors have to merge in order to activate the same response. This means that there are multiple external requirements that must be met before a cell commits to a specific response. The effects of extracellular signals can also be amplified by enzymatic cascade. At the initiation of a signal ligand binds to a single receptor. However, activation of receptor-linked enzymes can activate many different copies of a component in a signaling cascade, which amplifies the signal, showing you just how complicated these processes can become. Here's an example of a very common and very complicated cell signaling pathway. The epidermal growth factor receptor is a receptor tyrosine kinase involved in the regulation of cell growth, wound healing, and tissue repair. When epidermal growth factors bind to their receptors, a cascade of downstream events causes cells to grow and divide. When the epidermal growth factor receptor is activated at inappropriate times, uncontrolled growth, cancer, can occur. In this image, what you're seeing are all of the different proteins and enzymes that are working together. Upon binding the epidermal growth factor to the epidermal growth factor receptor, two different proteins that are associated with that receptor, known as GRB2 and SOS, activate yet another protein, commonly known in cancer pathways as RAS. RAS works with the protein kinase. Remember, kinase means enzyme, called RAF. RAF is then going to turn around and share phosphorus with something called MEK or MEK, which then turns around and shares yet another phosphorus with something known as ERK. ERK is a MAP kinase. The phosphorylated ERK enters the nucleus where it triggers a cellular response. ERK is actually going to stimulate cell proliferation, cell migration, and adhesion angiogenesis, which means that new packet of cells is going to grow its own blood vessels. It's also going to inhibit apoptosis, or normal cell death. This is an example of a receptor tyrosine kinase leading to the proliferation and growth of a cell regardless of if it's actually healthy for the system. There are a lot of methods of intracellular signaling similar to the one that you just saw. The induction of a signaling pathway depends on the modification of cellular components by an enzyme. There are numerous enzymatic modifications that can occur, and they are recognized in turn by the next component existing downstream. Two common methods of intracellular signaling are phosphorylation, which we've just seen a lot of, and molecules known as second messengers. One of the most common chemical modifications that occur in signaling pathways is the addition of a phosphate group. The phosphate can be added to a nucleotide such as GMP to form GDP or even GTP, and phosphates are often also added to particular amino acids on proteins where they replace the hydroxyl group that normally sits there. This process is catalyzed by enzymes known as kinases. Various kinases are named for the substrate that they phosphorylate. Phosphorylation of serine or threonine residues often activates enzymes and is commonly studied. Phosphorylation of tyrosine residues can either affect the activity of an enzyme or create a binding site that interacts with downstream components of a signaling cascade. Phosphorylation can activate or inactivate enzymes, and the reversal of this process is known as dephosphorylation. And it happens by something called a phosphatase. It reverses the effect of whatever the phosphorus was doing. Second messengers, on the other hand, are a little bit different than phosphorylation. Second messengers are small molecules that help our cells to propagate signals. A very common second messenger is calcium. Its concentration is traditionally low when we store it in vesicles in the smooth endoplasmic reticulum. Ligand-gated channels let calcium in, and the response can vary greatly. In the pancreas, it will cause the release of insulin, while in the muscles, it can cause contractions. Cyclic AMP, or cAMP, is a common second messenger as well, and it's synthesized by adenyl cyclase from ATP. It can bind to and activate a variety of cAMP-dependent kinases. That means that there are a lot of enzymes that are literally just waiting around for the cAMP to turn them on. cAMP is known to regulate vital metabolic pathways, and it activates a number of important proteins. The third type of second messenger we're going to learn about are inositol phospholipids. These have very small concentrations naturally, and they exist as membrane components. The most common is called PI, and we'll talk about it in just a moment. The picture you're looking at now shows you the mechanism for the formation of cAMP. cAMP serves as a second messenger to activate or inactivate proteins within the cell. Termination of a signal occurs when an enzyme called phosphodiesterase converts cAMP back into AMP. I mentioned those inositol phospholipids that can be converted into second messengers. They are normal membrane-bound components, so they're located near membrane-bound receptors, and therefore can very easily interact with them. PI is the main phospholipid that plays a major role in cell signaling. Enzymes known as kinases phosphorylate PI to make something called PIP and PIP2. The enzyme phospholipase cleaves PIP2 into two separate molecules called DAG and IP3. These products of cleavage from PIP2 serve as very special second messengers. DAG remains in the plasma membrane and activates a protein kinase known as protein kinase C, or PKC. PKC can then go about phosphorylating serine and threonine residues. We've already learned about those. IP3, however, diffuses into the cytoplasm, and it can bind to various ligand-gated calcium channels in the endoplasmic reticulum, leading to the release of calcium, and that can help to continue a signaling cascade. So we've looked at the types of signaling, we looked at who receives the signals and who sends the signals. Now let's take a look and see how we respond to all of this signaling. One response is an increase in cellular metabolism. Activation of specific receptors in muscle cells might lead to the increase of cAMP inside of that cell and therefore communication and movement within it. One response might be cell growth. Ligands called growth factors can be linked to tyrosine kinases referred to as RTKs. Once activated, they lead to cellular division. Sometimes a response can be termination of a signaling cascade, which is just as important as starting one. We can degrade or remove a ligand, and once we've done that, the effects are long-lasting. A major response is gene expression, and another very common response is apoptosis. Gene expression is the regulation of transcription of RNA or the regulation of translation from mRNA into proteins. There are numerous examples of signaling pathways altering gene expression, and one pathway is seen here, and it involves some players that we've met before. ERK is a MAP kinase that activates translation when it's phosphorylated. ERK phosphorylates something known as MNK1, which in turn phosphorylates something else, which is actually an initiation factor. The initiation factor is associated with mRNA, and it allows mRNA to unfold and begin the process of protein synthesis. When a cell is damaged, superfluous, or even potentially dangerous to an organism, a cell can initiate a mechanism to trigger programmed cell death, and this is known as apoptosis. Apoptosis allows cells to die or be disassembled in a controlled manner that prevents the release of potentially damaging molecules from inside the cell. There are many internal checkpoints that monitor a cell's health, and if any abnormalities are observed, a cell can initiate this process. Here's a histological section of a foot of a 15-day-old mouse embryo visualized using light microscopy. It reveals areas of tissue between the toes, which apoptosis will eliminate before the mouse reaches the full gestational age of 27 days. This happens in human beings. Our fingers and toes are webbed too, and some people are actually still born with that webbing. We even have tails that are reabsorbed back into our bodies. Communication in single-cellular organisms are a little bit different. Yeasts are eukaryotes, but they're fungi, and the components and processes found in yeasts are similar to those of cell signal receptor molecules and multicellular organisms, but still a little bit different. In order to find another haploid yeast to mate with, budding yeast cells will secrete a signaling molecule known as a mating factor. When the mating factor binds to the cell surface receptors of a different yeast cell that just so happens to be nearby, they will actually stop their normal growth cycles and initiate a cell signaling cascade that includes protein kinases and GPT binding proteins that are similar to G proteins. The yeast can then choose to share genetic information with one another to increase their offspring's variability and give them a better chance of survival. Signaling in bacteria is different as well. It enables bacteria to monitor extracellular conditions, ensuring that there are sufficient amounts of nutrients, and also to ensure that hazardous situations are avoided. There are even circumstances when bacteria communicate with each other. This has been observed in bacteria that have a symbiotic relationship with a Hawaiian bobtail squid. When the population density of the bacteria reaches a certain level, specific gene expression is initiated. The bacteria produce bioluminescent properties that emit light. This signaling is known as quorum sensing. Quorum sensing uses autoinducers to signal molecules. Autoinducers are signaling molecules secreted by the bacteria to communicate with other bacteria of the exact same kind. These secreted autoinducers are small hydrophobic molecules known as AHL, or larger peptide-based molecules. Each type of molecule has a different mode of action. When AHL enters a target bacteria, it binds to some transcription factors, which then switch gene expression on or off, depending on the situation. The peptide autoinducers, however, stimulate a more complicated signaling pathway. The changes to bacteria following exposure to autoinducers can be quite extensive. Cell-to-cell communication allows situations like this to happen. In A, Staphylococcus aureus bacteria have worked together to form a biofilm inside of something like a hospital patient's catheter, seen here via scanning electron microscopy. Staph aureus is the main cause of hospital-acquired infections. In B, you see that Hawaiian bobtail squid. They have a very symbiotic relationship with the bioluminescent bacteria previously discussed. Luminescence makes it very difficult to see the squid below because it effectively eliminates its shadow. In return for the camouflage, the squid provides food for the bacteria. So two organisms are capable of communicating with one another even though they don't hold anything that we would consider language.